Today's lecture deals with the interaction between a fetus and the mother. In other words, we're going to be talking about those connection devices called placentas. And they are wide and varied, at least 5 different types that we're going to go over. They are highly important because they are the sole chemical communication system point between a mother, her bloodstream, the food that's there, the oxygen that's there, and the bloodstream of the embryo or fetus, depending on when we're talking about, up to 11 weeks embryo, after 11 weeks to term fetus. All right? And in order for us to understand what's going on, we've got to talk about the extra embryonic membranes that are common to almost all embryos, not all, but most. 2 of them come from splanchno pleura and 2 of them come from somatopleur. In figure A, we see the 4 extra embryonic membranes. The 2 that I have in a brownish red here are the amnion and the chorion. The amnion around a human embryo is a sac in which the embryo floats in fluid, this blue area right here, called amniotic fluid. Now, in order for birth to occur, that sac is going to have to rupture, and that's what we call when a woman's water breaks. As I have said in previous lectures, that means that birth is imminent when a woman's water breaks. If it occurs very early, it's dangerous. She could very easily get an infection and lose the embryo, lose the fetus rather. Okay, so the chorion, which is a connection device part of the placenta, and the amnion, which is a sac containing fluid, amniotic fluid around the embryo, those are both somatopleur. What is somatopleur? Somatic mesoderm, the top layer of lateral plate mesoderm, and the overlying ectoderm fused together. Now, the other 2 extra embryonic membranes that we need to have a working knowledge of and be able to talk about are the yolk sac, which is outlined in yellow here, and the garbage dump called an amnion, which comes out here. Now, the amnion, in those beasties that are egg layers, okay, an egg layer like a reptile or a bird or some fishes. The amnion is a garbage dump. We've talked about that before. But in a human, the waste material is transported across the placenta to the mother, and it never does really pile up and accumulate inside the amnion in a mammal, in a human. So the amnion is transitory in us, in live bearers, okay, or not egg layers, but live bearers. Oviparous, meaning egg layers. Viviparous, V-I-V-I-P-A-R-O-U-S, meaning live bearers. And of course, humans are live bearers. They don't lay an egg in a nest. The most important part about the amnion is its wall is where a pair of the most important blood vessels that are in this whole shooting match right here, called the umbilical arteries and veins, develop. And they lead out to these finger-like projections on the surface of the placenta called the chorionic villi. Okay, blood goes out through these veins, arteries and veins, and it interacts with maternal blood that's on the outside out here. And we're going to talk about 5 different types of interaction that take place here. It gets complicated, but maybe I'll be able to help you to understand it a little more simply. So these are the 4 extra embryonic membranes, and you find them in most places, not in frogs. Frogs don't have all of these extra embryonic structures, but almost everything that lays an egg or delivers a live bear, a live embryo, does have these 4 extra embryonic membranes. Oviparous egg layers and viviparous, V-I-V-I-P-A-R-O-U-S, live bearers. Okay, The placenta is what we're going to be talking about today, and it is the sole connection point between the mother and her circulatory system and the fetal circulatory system of the embryo or fetus that's growing down inside her uterus, the placenta. The placenta has about 5 functions. First of all, it is a source of nutrition. It has a nutritive function. Nutrients, food molecules that are in mother's blood have to go across the barriers of the placenta into the bloodstream of the fetus, and there mother provides nutrients to the fetus as it grows. It's a respiratory organ. Oxygen goes from mother's blood to fetal blood, and carbon dioxide goes the other way from fetal blood across the placenta. Across the placenta, yeah, that's the way to put it. It is a barrier across the placenta to the maternal circulatory system. Okay? It's an excretory system. Nitrogenous waste products aren't stored in humans in the amnion, in the allantois, like they would be in an oviparous beastie. Instead, nitrogenous waste products are passed from the fetal blood across the placental barrier, the tissues that make up the placenta, into the blood system of the mother. Okay? And that then is the way that excretory products are gotten rid of by the fetus. They go into the mother's blood, her kidneys filter them out, she urinates them away, and that's the end of that. Okay, what else? It's a nutritive function, food goes across, respiratory function, oxygen goes from maternal blood over to fetal blood, carbon dioxide, higher concentration in fetal blood, so it goes across from fetal blood back to maternal blood. And then it is also a protective isolation device. And in order for us to understand that, I have to talk about something called Erythroblastosis Fetalis. And for that, we're going to use these 2 little comical drawings here. There's a husband and wife. Billy Bob is the father, and Sally Sue is the mother, and she is carrying the fetus, Molly Fred, down here. Okay? There is a thing that used to really take a lot of a heavy toll on human pregnancies. It caused a lot of them to end prematurely, and it killed the fetus, is what it did. It was called the RH syndrome, RH factor syndrome, or Erythroblastosis Fetalis. We see that the tissues that make up the placenta act as a barrier against things that might be in mother's blood that would attack something in the fetal blood. And the perfect example is Erythroblastosis Fetalis. We've learned to get around that now in the past 30 years, but for a while there was nothing you could do. If you were RH incompatible, you had to watch out. You might have one child, okay, but you never would have a second or especially a third, because they would die in utero and would be stillborn. They would not live. So here's what happens. There are several different kinds of components that are found in your blood. There's one family of proteins called the alpha globulins. There's a second one called the beta globulins. And then there's the really important ones that everybody knows about called gamma globulins, the third family of proteins that are found in blood. We know about type A blood that has a substance in it we call substance A, and type B blood, which is a substance that's different from A called B. Yeah. There is another, though, factor that is found genetically produced in individuals called the RH factor. It was first discovered in a monkey called the Rhesus, R-H-E-S-U-S, the Rhesus monkey. And I got to looking around, and lo and behold, it turns out that 85% of Homo sapiens, human beings, also produce this Rhesus factor, or the RH factor for short. The RH factor, if you have it, you're called RH positive. If you don't, you're RH negative. If you look at a blood type in shorthand, the way it's written up on a hospital chart, they'll say this patient has type B positive blood. What does that mean? It means they don't have type A substance, but they do have type B substance on their red blood cells. And they also have this RH factor. That's the positive. 85% of Homo sapiens are positive, RH positive, 15% are not. And it's dangerous. Thank goodness the placenta is a barrier. In addition to being a communication device, it prevents some things from getting from maternal blood over to fetal blood. And one of the most important things it prevents is the RH factor. Now, you either are RH positive, meaning it is normal for you to make this substance, this RH, this Rhesus factor, or you're RH negative. And it turns out, I'm going to tell you an illustration that is highly oversimplified. It's a lot more complicated, but I want you to get it easily here. So I'm going to give you the simplified version. The Rhesus factor, you can either have the genetic capability of making this factor, you inherit it from mama or daddy or both, in your genetic makeup, in your chromosomes, in your genes. And if you inherit the recipe for making the RH factor, you will make it, and you will put it on all of your erythrocytes, and it'll float around inside your blood serum, and it will be attached to other cells in your body. And you will be said to be RH positive. You inherited it from mama or daddy. Somebody in your ancestry was RH positive and passed that gene on to you. Now, this is the part that's oversimplified. It is as if your body, the day you were born, or maybe right before you were born, your system called your immune system, the system that protects you from foreign germs and foreign bacteria and foreign proteins that might get into your body. It's as if your immune system, boy, I'm oversimplifying, sort of took an inventory the day you were born and looked through your whole body and checked off on a list, boy, am I oversimplifying, and said, this protein belongs here. It's in the brand new born little infant, so it's self. It belongs there. It's not foreign, okay? It's self. And your body sort of makes an immune list of things that belong, which are called self-proteins. And if you are supposed to have this protein, this RH factor, then you will know about that from the day, your immune system will know about it from the day you were born, okay? But your immune system sort of keeps this inventory list all your life, and any time during the rest of your life, if a foreign protein gets into your body, let's say a COVID virus protein, yeah, that's not something that was there the day you were born. And it's not self, it's foreign. And your body, your immune system will say, that's not supposed to be here. Let's attack it. And gets all the other immune competent T and B cells together, and they attack this foreign protein and build up an antigen response, a response made up of antibodies that attack that one specific foreign protein and sweep it out of your body and get rid of it. Foreign proteins, your body will react against and will try to get rid of. Self proteins, no big deal. You're allowed to just live with it because it's supposed to be there. All right, the Rhesus factor, maybe when you were born, You didn't get the gene from either mother or daddy for the production of this protein, this Rh factor. And so, you never did have it on your blood cells. It was not self. You were Rh negative and would be for the rest of your life. Yeah. What if you inherited the gene for making this Rh protein from either mama or daddy or both? Well, that meant when you were born, your red blood cells had this little protein stuck on the outside, and it was there from the day one that you were born, all the rest of your life, and it would be known as a self, not a foreign, but a self protein. And your immune system would never, ever attack it and try to get rid of it like it will attack a germ or something that's not supposed to be inside of you, some foreign protein. Well, it turns out that you got a problem. If Papa is Rh positive and he gives the gene to Mama in his sperm that combines with her egg to form Molly Fred, right here, Molly Fred may be positive, Rh positive, have Rh positive blood. Type A positive or B positive or AB positive, but he'll have positive if the gene came from Papa to Mama. All right, that's foreign protein right here. Now, it turns out that if Mama is Rh positive also, just like Papa was, well, there's no problem. Down here, if both mother and father are Rh positive, then there is no way that Mama's blood will attack the blood cells inside the fetal body of little Molly Fred. It won't happen. No, there's nothing foreign there. But what if, and this is the bad one, what if Mama is Rh negative? All right, and to her, the Rh protein is a foreign protein. If it ever comes into her body, her immune system will attack it and try and get rid of it. Well, what if Billy Bob is positive, and he gives that positive gene to Molly Fred, and Sally Sue then is Rh negative? That means that there are proteins, antigens, okay, down here, or foreign proteins that make up the body and the blood of little Molly Fred that are foreign to Mama. Oops, all of a sudden we have something that shouldn't be in her body that her immune system will attack. And so, usually during the first pregnancy, if she has multiple pregnancies, the placenta acts as a barrier to prevent her antibody-producing cells that would attack the fetal blood tissue down here, the positive blood tissue. Most of the time, the placenta protects Molly Fred until Molly Fred is born. But when Molly Fred is born, there is mixing of maternal blood and fetal blood because there's a stripping away of the layers of the placenta, and blood will mix from placental fetal blood over into Mama's blood system, and her body will go on a witch hunt looking for those nasty old Rh positive blood cells that got into her blood system when Molly Fred was born. And for the rest of her life, anytime an Rh positive protein shows up in her body, her immune system is going to attack it and try and kill it. So what about the second pregnancy? Let's assume Billy Bob is positive and he gives the gene to the second pregnancy, Molly Fred Jr., number 2, or whatever. And mother up here now is sensitized. Her immune system is ready to attack any cell that has an Rh positive protein on it. So her immune system is now primed and ready to go and loaded for bear, boy. It's going. And oftentimes it'll go right across that placenta. The placenta is just not that strong a protective device for a second pregnancy, especially with a high antibody, anti-Rh antibody level, titer it's called, in Mama's blood system. And eventually, her immune competent cells, B and T cells, will attack through the placenta and attack the tissue of the second pregnancy and kill it. So, used to be 40, 50 years ago, before they came up with a way to get around this, it turned out that if you were going to get married, you had to go, before you could buy a license, you had to go get some tests done from your local doctor for venereal diseases, for one thing, but also for an Rh incompatibility. And they would tell you right off the bat whether or not there might be the problem of a second pregnancy being subjected to possibly being destroyed. And that would only happen if Papa's Rh positive and Mama's Rh negative. Down here, if Mama and Father are both positive, no problem. Why? Because the Rh factor to her, if she's positive, is not a foreign protein. So she's never going to make antibodies to attack her fetus that's also, okay? What if Mama and Daddy are both negative? Well, if he's negative, he can't give the gene to this fetus, to little Molly Fred, and so little Molly Fred will never be AB positive. It will always be something negative. And of course, if it's negative, then Mama's blood will never attack it. So, if both mother and father are positive or mother and father are negative, no problem. No erythroblastosis fetalis, which is Mama's blood attacking the tissues of the fetus. What if mother is Rh positive, father is Rh negative? It doesn't matter. If she's positive, her body already knows about that protein. It's not foreign. It was on her first day inventory when she was a brand new little, and she will never make antibodies against it, so there won't be any antibodies to attack her fetus, even the second, third, fourth, fifth, any of them that she has. But the condition where it's a problem is when Billy Bob, okay, is positive and Molly Fred is negative. Then that could lead to a lethal condition. It could kill the fetus, usually not the first one, but any subsequent second, third, fourth pregnancy, it would lead to probably death long before birth. And this Molly Fred would be killed in utero and not be born alive. Erythroblastosis fetalis, better known as the Rh syndrome. Nowadays, we have a way around this. Let's suppose we do have a problem. He's positive, she's negative. Just as soon as she gives birth to her first child, Molly Fred, who happened to get the Rh positive protein from Papa, we give her a shot of something called Rogam, that's the commercial name, passively acquired antibodies that sweep through her body the 10 minutes after she gives birth and looks for every fetal blood cell that got out into her body and destroys it so it never has a chance to ever build up an immune concentration, a titer, or an immune response to those little positive cells that came from the placenta when it peeled apart at the time of birth. Okay, so now we can give a mama a shot of Rogam, and that helps her to be able to have a second, third, possibly fourth pregnancy without a complication. All right, we got a way around it. But all of that is to explain the fact that erythroblastosis fetalis is lethal, and points out the fact that the placenta is not only a communication device, allowing things to go from mama's blood to fetal blood and vice versa, fetal blood to mama's blood, it's also a protective device in that it prevents some things from getting across the placenta from maternal blood, such as antibodies against the Rh factor, from mama into fetal blood where it can do harm. Okay. All right. Let's see. We want to talk about the tissues that are found in a placenta. And there are at least 5 types of tissues that have various combinations of these tissues that make up the placental barrier. That is, here's maternal blood found in a mother's blood vessel inside her uterus. Here's fetal blood found in a blood vessel inside a growing fetus or embryo up inside her uterus. But between those 2 blood systems, there are going to be 1, 2, 3 tissues that mama made, which act as a filter, a barrier, to transplantation, to something going from mama's blood across the placenta to fetal blood, or vice versa. And there are going to be 3 types of tissue in the fetal tissues that also are barriers to placentation, 4, 5, and 6. And so we're going to look at 5 different kinds of placentas that mammals have. We're going to name them in 2 ways. One is which of the maternal tissues is the outer maternal tissue, and which of the fetal tissues is the one that plasters up against it, is up against the maternal tissue. Which of the fetal tissues is the outer one next to the outer fetal maternal tissue. Yeah, and so we'll start out with the one that is the most, has the most number of tissue barriers. First of all, let's look at what we're dealing with here in, say, a human. Here is an embryo. It is floating in a sac called an amnion, full of amniotic fluid, okay? And coming out of the umbilical cord, there's a blood vessel, 2 actually, and they go out into these chorionic villus. If you don't remember what that is, go back to the last lecture where we talked about the chorion and the different tissues that make up these invasive little finger-like processes. Okay? Now, it turns out that in a human, these little finger-like processes, which are made up basically of 3 different kinds of tissue, they are bathed by maternal blood. There aren't any maternal tissues that act as barriers to transplantation. Maternal blood bathes these little chorionic villi that stick out up in the placenta, inside the uterus. And so stuff can go, say oxygen can go from the blood of mama out here to the blood that's on the inside of this blood vessel right there by going through the outer chorionic ectoderm, this outer membrane around the villus, this finger-like projection, and then on the inside of that villus, that hollow finger-like projection, there is some connective tissue, and the oxygen has to go through that, and then it has to make its way through the fetal blood vessel wall, a 3rd barrier. 3 barriers to transplantation for a molecule of oxygen, but once it's gone through, boom, 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 all 3 of those barriers, it gets into the blood of the fetus, and the oxygen is taken into the embryo, and that's the way the embryo gets its oxygen. It can't breathe, has to rely on mother's blood. So there are 3 barriers to transplantation in this type of fetus right here. But that's one of the middle types. Now, what I want us to do is to look at all 5 major types of placentas now. And we're going to start with, by far, the most complicated one. This one, in figure C, has 6 barriers to transplantation. Let's look at them. The 6 barriers. Let's talk about a molecule of oxygen that we want to get from mama's blood inside a blood vessel up inside her uterus here, her placenta. We want to get through all the tissues that are of maternal origin and get through all the tissues that are of fetal origin up on the inside of this chorionic villus. Okay? And eventually that molecule will go from the hemoglobin here to the hemoglobin in the blood that leads down into the embryo. 6 barriers. And the outer barrier of maternal origin is this outer folded layer right here of uterine epithelium. Okay? Inside of that, there is this layer right there of connective tissue of the uterine endometrium, endometrial tissue right here. And inside of that, there's the third barrier, which is the wall of the maternal blood vessel right there. So we got 3 barriers of maternal origin to transport of an oxygen molecule down on its way to a fetal blood vessel. Then there are 3 more barriers of fetal origin, and they are the 3 barriers associated with this long invasive villus. There's the outer ectoderm right here. That's the first of the fetal barriers. Then there's the connective tissue up on the inside up in there. That's the second barrier of fetal origin. And finally, the wall of the fetal blood vessel is the third barrier to transplantation. Well, 6 different barriers an oxygen molecule has to go across, get through, when going from mama's blood to the blood that goes to the embryo right here. That is a really, wow, a really complicated system. Now, if we were to just dissect out the placenta, just the parts that came from the fetus, not from mama, and look at these chorionic villi, these finger-like processes, the placenta would look like this. It would have 6 barriers to transplantation, and it would have villi equally scattered all around on the surface, these little poked out finger-like processes of fetal origin. Now, that's called a diffuse, D-I-F-F-U-S-E, way of arranging the chorionic villi on the surface here. So this is sometimes called a diffuse placenta. The other technical name for it uses the name for the outer maternal layer that is in contact with the outer fetal layer. And there's a better way for us to look at that and come up with that name. So here's what I'm talking about. This is the way we get the second name. We said that it was called a diffuse, and that's the way that the villi are arranged on the surface. But notice that the outermost layer of maternal origin tissue that oxygen molecule has to go through is the uterine epithelium on the outside. And the shorthand prefix for that is epithelio hyphen. The outer fetal origin tissue that the molecule of oxygen has to go through would be the outermost chorionic ectoderm on the outside of that finger-like projection, and that's called the Chorionic Ectoderm of the villus. So epithelio, and the shortened term for the Chorionic Ectoderm is /Chorion. So epithelial coil is the second name for this type of placenta. This right here. It's an epitheliochorial or diffuse, and it has, count them, has 6 layers that transplantation has to go through. 1, 2, 3, that's the outer maternal tissue. 4, 5, 6, and that's the outer fetal tissue. So we use these 2, this prefix and this suffix, as the second name, the epitheliochorial type. And it is found in pigs and horses, in case you're wondering. Oftentimes when a mare gives birth to her foal, to her baby horse, there's no blood because the tissues just peel apart. And there's very, very little bleeding, if any. All right, that's the first type of placenta. Here's the second type of placenta, and it gets more simple. It gets more simplified. This one has lost one of the barriers to transplantation. Whoops. Let me get this up here. Here we go. Here's the maternal blood vessel. Then here is the endometrium tissue up inside there. But the outer layer of endometrium tissue, the villi have poked through it. It's no longer a barrier. This epithelial tissue right here is just not a barrier anymore. There are only two barriers of maternal origin to transplantation: the wall of the maternal blood vessel and this layer of endometrial tissue right here. But look, all three layers of the fetal placenta, the outer chorionic villus, ectoderm here, the Syndesmo Corial, fibrous tissue that's on the inside up here, and finally the wall of the fetal blood vessel. All three of those barriers are there. What is the one barrier that we have eliminated? It's this barrier right here, the uterine epithelium. It's no longer a barrier. Also, we can name this not only according to which maternal tissue and which fetal tissue are in juxtaposition, or the outside ones. We can also talk about the arrangement of the villi on the surface of the placenta. And it turns out that they are in little patches called cotyledons. Now, I know some of you may have heard the word cotyledon if you've studied botany, because that also is a part of a seed. But that's not what we're talking about now. These patches of villi on the surface of the placenta are called cotyledons. So this is sometimes called a cotyledonary type of placenta. And it is found in ruminants, pigs, cows, sheep, ruminants. They have this 5-barrier transplantation type of barrier in their placentas. So we have the cotyledonary or the Syndesmo Coil. Where in the world did we get Syndesmo? Well, Syndesmo is the word that we use for connective tissue. So let's look and see what's missing here that was found in the first barrier, the first type of placenta. What about the tissues of maternal origin that an oxygen molecule has to go through? Well, the wall of the blood vessel is still there. The connective tissue up in the uterine endometrium is there. In connective tissue, we use the prefix syndesmo to refer to connective tissue. All right? But the uterine epithelium is no longer there because the villi have poked their way through it. It's no longer a barrier. But all three layers of the villus is still there. The outer chorional ectoderm is still there. The connective tissue up inside it is still there. And the blood vessel wall of the fetal blood vessel up inside the villus is still there. So this layer, the chorionic ectoderm, which the suffix is Corial, something Corial, is adjacent to this layer of maternal tissue, this connective tissue. Syndesmo Corial is the name, or cotyledonary if we're talking about the shape, the way that the villi are arranged on the surface. Syndesmo Corial, and that's in ruminants, cows and sheep. Okay. I told you there are going to be 5 of them. They get more complicated as we go along. Here's another, the third type. This type of placenta right here gets rid of another layer. There are going to be only 4 layers of transplantation barriers. Notice that the villi not only have poked their way through the uterine epithelium, mama's tissue, they've also plastered all the way up until they touch the wall of the maternal blood vessel. In other words, this layer of connective tissue in the in the endometrium is no longer a barrier. There is only one barrier to transplantation of maternal origin, and it is the wall of the maternal blood vessel. And so that's the word we use for that is endothelial. Epithelial is a covering tissue. Endothelial has to do with the wall of a blood vessel. Okay? But look, all three layers of the chorionic villi are still there. The outer ectoderm, the inner stuffing, and the wall of the fetal blood vessel. All three of those barriers to transplantation are still there. So, oh, by the way, if we look at the arrangement of the little finger-like projections, villi on the surface, they form a zone or a band around here, like that. Yeah. And so this is called a band type of placenta. It's found in dogs and cats mainly. And the other way that we can name it is, what's the outer layer of maternal tissue that's a barrier to transplantation? It's the wall of the maternal blood vessel right here, okay? And we said that the word for that is endothelial. And of course, the outer layer of Chorional Ectoderm on the outside of the villus here, the name for that is Corial. So this is a band type or an endothelia Corial type, according to which tissues are in contact up there. It's found in cats and dogs. And let's take a look at what we have eliminated to get this epitheliochorial name. What is the maternal outer layer of tissue? It's the wall of her blood vessel. And the word for blood vessel wall, the prefix is endothelial. But the connective tissue stroma is no longer there, and the uterine epithelium is no longer there. They're gone. They've been punctured by the villi sticking up. The tissues of fetal origin, all three of the chorionic villus tissues are still there. And the outer one is the chorional ectoderm, and that's the suffix corial. Endotheliocorial is the name of this type of blood vessel. Or it also has that band of villi around it, a zone or band. So it's called the band type. And it's found in cats and dogs for the most part. That's the most common way we have of finding one. Yeah, so it's getting simpler and simpler as we go, isn't it? Now we're going to get to the type that humans have. Oh, guess who else besides humans has this type of placenta? I don't mean to shoot down your opinion of yourself, but rodents, rats. Humans and rodents have this type of placenta. In this placenta type, first of all, humans have only one patch of chorionic villi. It's over on one side, okay? This little disc of villi is called a discoidal placenta right here, over on one side. And the big fancy name for it is there aren't any barriers to transplantation of maternal origin. The wall of the maternal blood vessel leaks out into a pool of blood up inside mama's endometrium, and her villi stick up into the pool. So there are those three layers, the wall of her blood vessel, the connective tissue of her endometrium, and the outer epithelium of her endometrium. Those are no longer barriers. They're just not there. Now, the prefix for blood is hemo, H-E-M-O dash. Okay. And the outer layer here is the chorional ectoderm, so corial. This is a hemocorial or a discoidal type of placenta with only how many barriers? They're all embryonic. 1, 2, 3 barriers. So let's take a look at what we've gotten rid of. Bet you're glad that I furnished these for you. Okay, here's the way we can explain what's going on. All the tissues of maternal origin are now no longer barriers. They're gone. So we have the fetal finger-like villi poking up into a pool of maternal blood. So the prefix for this type of placenta is blood, hemo, and the outer layer of fetal origin is the chorionic villus or Corial. Hemocorial is the name of this type of placenta, named according to which tissues are into intimate contact of maternal and fetal origin. It's also called the discoidal because it had one disc of those villi stuck out on the side. Okay. Now, I would like to show you a pretty picture of a rabbit, the fifth type of placenta, but I can't because there is no picture. Here's the way it works for a rabbit, which has the most simple type of placenta that there is. Nothing but the wall of a fetal blood vessel, that's its outer fetal tissue, is in contact with no tissues of mama, only her blood. So this one, it doesn't have a villus. We can't say that it has a disc of villi or a band or something. There's no way of naming this particular thing. It's found in rabbits and lagomorphs. Rabbits and lagomorphs. A lagomorph is a rabbit. Okay. And the maternal prefix is hemo, meaning that the blood vessel is completely surrounded by maternal blood. And the only thing that is a barrier to transplantation is the wall of the maternal blood vessel sticking up in the middle of what used to be a villus, and that's called the endothelial. This is a hemo-endothelial barrier, one barrier to transplantation of something going from maternal blood to fetal blood, or waste products going from fetal blood to maternal blood. There's only one tissue barrier. This is the fifth type. It doesn't have villi, so there is no villi name for this type. Now, I will provide for you this summary sheet to help you to figure out what's going on here. The 5 types of mammal placentas. They are in order: 6 barriers, 5 barriers, 4 barriers, and 3 barriers, and 1 barrier. Here, in order for, this is the most extensive, horses and pigs, the most extensive protected transplantation safe interaction between maternal blood and fetal blood. Maternal blood is inside a maternal blood vessel, so the wall of the blood vessel is a barrier to transplantation. Then there's this material that makes up the endometrium in here, the connective tissue around the maternal blood vessel up in the uterus. And finally, there is a layer on the outside of the endometrium of uterine epithelium right here. All 3 of those are barriers to transplantation. What about barriers now they're maternal origin. They're made by mama. What are the ones that are made by the fetus? Well, they are the 3 layers of the chorionic villus. The outer ectoderm, chorional ectoderm of the villus, the connective tissue sticking up inside that finger-like process, and finally the wall of the fetal blood vessel. 1, 2, 3, 4, 5, 6 barriers to transplantation. Whoa, it's the epithelio-corial. It also has diffuse all over the place, villi all over the whole surface. It's in pigs and horses. Get rid of this barrier right here, so that this is pressing up against this. And that's what we've got, the second barrier right here. That's 6 barriers to transplantation. Here's the second type with 5 barriers. There are only 2 barriers of maternal origin. This stuff right there endometrium tissue, and the wall of the maternal blood vessel. Those are the only barriers of maternal origin. But the villus still has all three of its barriers. So 2 and 3 is 5 barriers here. And these are found in ruminants, sheep and pigs and cows. Yeah. And this is called the Syndesmo Corial. Corial because the Chorional Ectoderm is the outermost fetal layer. And Syndesmo means this connective tissue material up in here is the outermost maternal tissue, Syndesmo Corial. Then when we get rid of that tissue right here and get to the third type of placenta, we have only 4 barriers. The wall of the maternal blood vessel is pressed right up against the outer ectoderm of the chorional villus, right there. So we got 1, 2, 3 barriers of fetal tissue and 1 barrier of maternal tissue. Yeah, and the barrier of maternal tissue is the endothelial wall of the blood vessel. So it's an endothelial and chorional ectoderm, Corial, endothelial Corial. And it's found in carnivores, cats, dogs, wolves, bears. Yeah. Woo. And then finally, if you go to 3 barriers, the wall of the maternal blood vessel is no longer a problem. The maternal blood vessel wall breaks, and what we have is just a bunch of villi bathed in maternal blood. Guess what? That's us, humans, and rats. I'm sorry. Anyway. And this is called a hemo blood, chorional ectoderm, hemocorial placenta. And we notice that the villi are in a disc in one area, a discoidal placenta with 3 barriers. And then finally there was that 5th type that we find only in rabbits and lagomorphs and hares. And it has lost everything but the wall of that one fetal blood vessel. And it is bathed by maternal blood. There's just one barrier to transplantation. The Rh factor can get across that real easy. Thank goodness rabbits don't have that problem. The Erythroblastosis Fetalis, it would kill them all. So that's the 5 types of mammalian placentas right there. Oh, complicated, huh? All right. That basically is the material that I want you to consider concerning the tissues and the physiology of the placentas that are found in mammals. And that's all for this particular lecture. I thank you very much, and good day.